Hello, and welcome to this Center for Biosimilars Peer Exchange, titled Understanding and Improving Access of Biosimilars in Oncology. I'm Dr. Bruce Feinberg, Vice President, Clinical Affairs and Chief Medical Officer at Cardinal Health Specialty Solutions. And joining me today in this virtual discussion are my colleagues, Dr. Karina Abdallah, Blue Cross Blue Shield of Michigan, Dr. Michael Diaz, Florida Cancer Specialist and Research Institute, Kathy Ube, Pontchartrain Cancer Center, Covington, Louisiana, and Bhavesh Shah, Boston Medical Center Health System. Today, we're going to discuss a number of topics pertaining to the use of biosimilars in oncology, including improving market access and the economic impact. So let's get started on the first topic. And we're going to give an overview of biosimilars. Now, I know this can be problematic for many of you. I first started speaking on the circuit on biosimilars 6 years ago. So for many, this is old hat. Been there, done that, but it begs a question to level set on the subject, because despite the awareness that many seem to have, it begs the question, why has the market uptake been so limited? And I think for that reason, it's worth going back a little bit and understanding the basic pharmacology of biosimilars, what differentiates them from generics. So we're not going to belabor this, but we're going to get into some of these basic questions so that it's clear to everyone on this call, but also clear why some of these issues may still be confusing to practitioners, and particularly in oncology, where the availability has been very limited. So what are the similarities and differences of biosimilars from biologic reference brands? Again, this difference in regard to generics, the naming that has gone through already changes and iterations for biosimilars. And so to get us started with that, I'm going to ask Bhavesh to kind of begin to give us that little bit of background and to give us some of those basic definitions and understandings. Bhavesh.
Sure. Thank you, Bruce. So obviously, there's a very basic definition that we as healthcare providers understand. Biosimilars are biologics that are similar to the reference product with no clinically meaningful differences in regards to potency, purity, and safety. And I think though the biggest aspect is that, trying to explain a patient that what a biosimilar is using that definition. So I see that as a gap, as a patient education gap. And then talking about other gaps in regards to biosimilars are interchangeability, which we know that it is one of those standards, which is a regulatory term. And a lot of times providers and patients think that there's a higher designation that it gives a biosimilar. And if it doesn't have that it doesn't actually function as same as a biologic. So there's definitely a lot of provider and patient education that we need to help with closing the gap. And then I think one of the other aspects that I still see people struggle with is that the FDA, I have providers come to me and say, how is it that the FDA did a phase 3 randomized controlled trial in one indication and then extrapolated to 7 different indications? So there's definitely this education gap in terms of the whole process, besides the definition from my perspective.
So I think, Bhavesh, you bring up some great points. So the first point has to do with the clinical trial design by which biosimilars gain their approval. So their comparison to the reference brand was often in a non-inferiority trial. So we have a trial design in of itself that certainly in the world of oncology is held more suspect. Then we have the issue of extrapolation across indications, which is also something that's anathema in oncology, whereas it may be accepted in inflammatory diseases to a greater degree. And then we've got this issue of interchangeability, in which it's problematic in and of itself because to some degree, interchangeability is difficult even for the reference brand because of the manufacturing process, which continues to have that product evolve. So the Rituxan that was manufactured 20 years ago may be very different from the Rituxan that's being manufactured today. So could you just maybe touch on that for a moment too?
Sure. So I think the manufacturing is a different aspect also, and another myth that I always heard is that a lot of rituximab, each lot of rituximab is a biosimilar of itself, and that's really not true because the manufacturing process is completely different. What's called product drift versus a biosimilarity process that's established for a biologic to go through in order to get approved as a biosimilar. There's actually critical quality attributes that the manufacturing process is established from a reference product. And basically the manufacturer has to meet those critical quality attributes in order for it to be go into production. So if it's out of that reference range, there's implications on the mechanism action, the potency of the drug, and those are the reference ranges that are established for manufacturing and not for a biosimilarity process.
So that's great, because that's the kind of answer that immediately gets people to like open their eyes and say, wow, this is more complicated. This isn't aspirin where 21 atoms are going to now be made by somebody else who's going to string together 21 atoms, and it's going to be exactly the same. These are the exact same chemical. There's no difference between them. You can call them something different, but it is what it is. And when you get into 20,000 atoms that are not identically strung together, then you have to look not at their chemical signature and identity, but more in some kind of a surrogate by which you say they are equivalent. And it becomes more about the process of manufacturing almost than it does the end product.
Whenever I'm trying to explain things to patients, as you alluded to, generic medications are a simple molecule that is simply made in a chemical process in a chemical plant. It's got one unique structure. These are chains of hundreds, if not thousands, of those molecules that are assembled by a living organism. And by its own nature, it's much more complex. There's all sorts of subtle differences that can exist and develop even in the same process over time. Not only that, whenever you're looking at it for an indication, and I'll use filgrastim as an example, there are many different indications for the use of this medication to stimulate the growth of white blood cells, but in each of these indications, they might be stimulated for different reasons. And so you're not only looking at different drugs, different biosimilars, you're also looking at different unique specific applications.